All right, today we're talking about cell types and the basic structures, such as some of the organelles. So let's dive in. We're going to start with how we organize cells based on their cell type and how they fit into the overall way that we classify life on Earth. So there's some learning objectives and our key terms. So just as a reminder from your previous video, cells are the basic unit of all living things. Just as we learned with the biomolecules how they all have a monomer, a building block, and we repeat that structure over and over to make the larger molecules, kind of similar to life. If you are alive, you must have at least one cell. You are at least one cell. And then we can always add more cells on top of that and you can become a multicellular organism, but you need to be a cell to be alive. So science has to develop a theory around what it means to be a cell and what that means exactly. So theories in science are very well tested hypotheses, meaning we see an observation, we make a hypothesis about it, like, in the case of Schleiden and Schwann, they're like, every single plant I look at and every single animal whose tissues I look at, they're all made of cells. So from there, they tested it so rigorously, not just them, but other scientists as well. We all tested it, and we can say with extreme certainty, based on all the evidence that was collected, all living things are made of cells. And it's a well-tested, well-proven hypothesis. And then we have the secondary part of cell theory, which is that every cell comes from pre-existing cells. As we can see in the image here, a cell will divide to form new cells. So those are the two core tenets of cell theory. So there are two types of cells on Earth. And the reason for that is basically this idea of compartmentalization. This really defines the differences between the two types of cells. When cells first evolved, they were literally just a water-based solution, so a bunch of chemicals dissolved in water, and then those became surrounded by a membrane, and they would perform coordinated chemical reactions. So they were very much like a studio apartment. There's one cell membrane, and they kind of have some chemicals inside, but otherwise it's all one big living space. And over time, these cells evolved more and more and more complexity, usually by compartmentalizing themselves. They would create smaller pockets within their cell structure to have more specific coordinated activities occurring. So let's look at one example here. DNA replication, that is an anabolic process, meaning we are building a new molecule. We're starting with something small and creating something larger at the end. Whereas digestion, we're doing the exact opposite. We're taking something large and we're breaking it down. Digestion often requires lots of enzymes that break molecules, lots of acid. We don't really want all those things around when we're doing something as important as DNA replication. So it would be nice if we could do DNA replication in one area and stick all of the acid and all of those digestive enzymes in a separate area where they won't interact with each other. That's basically how this started to occur. And eventually, each of these little compartments, became the organelles that we know and love, sort of. So let's actually give these names now. The studio apartment is more like a prokaryotic cell, whereas the more complex divisions of a home or a multi-bedroom apartment, that's like a eukaryote. So let's dive into what these two types of cells are. So looking at the term, pro means before, karyon means kernel. So this is before the kernel, or in this case, before the nucleus. A eukaryote, U means true, so this is the true kernel or the true nucleus cell. So prokaryotes, since they evolved before a nucleus evolved, They're smaller, they're more structurally simplistic, they do not have a nucleus, and they do not have membrane-bound organelles. Those two things are definitive of a prokaryote. Smaller, yes, that's true, but the most important thing to know is they do not have a nucleus, and they do not have any organelles that have membrane that makes them up. So they have some organelles like little ribosomes and other types of organelles that are not made of membrane. But any type of nucleus or mitochondria or ER, any of those things, they're all made of membrane, so these guys do not have them. And then eukaryotes, excuse me, are larger, much more complex because they have all those compartmentalizations occurring in there, those membrane-bound organelles, and one of those membrane-bound organelles happens to be a nucleus. So the main difference is eukaryotes have a nucleus, hence true nucleus and before the nucleus, and they also have those membrane-bound organelles. So prokaryotes are going to make up domains, two different domains, bacteria and archaea, which I'll get into that in a moment, whereas eukaryotes only make up one domain of life, which is eukarya. So this is basically all your bacteria, and eukaryotes make up everything else. Your protists, your fungi, your animals, your humans, your plants, anything else that's not bacteria is a eukaryote. So whether you are a prokaryote or a eukaryote, if you are a cell of any type, you must have 4 structures. So every single cell has at least these 4 basic things. Then they can add on to them. If they don't have these 4 things, they're not a cell. So the first one is the plasma membrane. Your cells are made up of a water-based solution. So how can you tell your water-based solution from the water-based solution next to you? You can't unless you have a plasma membrane, a barrier that's going to separate you from everything else in the world. Then we have the cytosol or the cytoplasm. That is the water-based solution, the water-based liquid that fills the cell. And this is effectively an aqueous solution. So all of our chemicals that make up the cell are dissolved in this water-based solution, and this is where all the chemical reactions of life occur. Then every single cell has to have genetic material, DNA, because this is the code that tells the cell what proteins to make, when to make them, how many to make, all of that kind of stuff. So it's basically the blueprint for the cell. You need to have the blueprint to know what to make and how to fill your cell. And then you have ribosomes. These are important because they are the organelle, the structure that can convert the DNA code into the proteins. So you need something to be able to read that DNA and turn it into a protein, and that's what ribosomes do. These are not made of membrane, so therefore all cells have them, whether you are a prokaryote or a eukaryote, since they are not a membrane-bound organelle. So when we look at the overall organization of life, we discussed all these larger portions in ecology. We've been discussing the atom and the molecule. Now we're getting into the cell, the organelle, and the tissue level a little bit, but mainly just the cell and the organelle. Now on top of that, we also classify life by type of life. So not just, oh, well, if you're the same type grouped together, we call you a population. If you are the same type of tissue grouped together, we call you an organ. We actually name organisms by what type of organism they are. And you guys know this. We have species names, scientific names that name a species for what it is. And that falls into a broader classification context. So species, genus, family, order, class, phylum, kingdom, and domain. So the domain one is really the only one that we're going to get into and care about in this class. So it's the highest level of organization, and it's based on what type of cell you have and what stage of evolution does that cell come from. So let's get into that. There are 3 domains of life. So every single living thing, as you classify it, will fall into one of 3 categories at the very beginning. The first one is bacteria. So these are prokaryotic cells, meaning they have no nucleus, they have no membrane-bound organelles, and they are always single-celled. So it's only one little cell living its own little life. It never has many cells put together like our body. And this is the oldest type of life. So the domain bacteria evolved first. Life evolved way back here, and at some point, the cell that we know as bacteria diverged from what eventually became the archaea and the eukarya, the other two domains. So this contains all of the bacterial species that we know today. Then there is archaea. This is also a prokaryote. So no nucleus, no membrane-bound organelles. It's very, very, very similar to bacteria, except if we look at this evolutionary timeline, it is more related to eukaryotic cells than it is to bacteria. It's still a prokaryotic cell, so the nucleus hadn't evolved yet. The organism that split off from it evolved a nucleus, but it did not. So it's more similar to bacteria in that way, but it kind of handles its DNA a little bit more like us. And there's a few other little characteristics it has that are a bit more similar to eukaryotes, but it is a true prokaryote. Now, a lot of times we would just look at an archaea and be like, oh, so it's a bacteria. They seem very similar. They are technically different, but for all intents and purposes for our class, it's just a single-celled organism that is very similar to bacteria. And then there is eukarya. This is the most recently evolved domain of life, and it is eukaryotic organisms only. So cells that have created a nucleus and membrane-bound organelles. Some of them can be single-celled, like our protists, like algae. Some fungi can be single-cellular. Otherwise, they're all going to be multicellular, like our plants and our animals. So a prokaryote has the more simplistic structure. Basically, what it is it's a cell membrane surrounding a kind of tangle of DNA. It's not truly a tangle. They do organize it a little bit, but it's a jumble of DNA in the middle. So we call that the nucleoid region. Wherever the DNA clusters, that's the nucleoid region, because they don't have a nucleus to store the DNA. They just have a region where the DNA is most likely going to be. And they only have one chromosome. So humans, we have 23 pairs of chromosomes. They only have one. It doesn't even have a pair. And it's usually almost always circular. There are a few that don't have circular chromosomes, but we don't really need to talk about them. They're weird. And then there's plasmids. So sometimes bacteria can do something cool. Prokaryotes in general can do something cool. They can take on really, really tiny circular pieces of DNA that are not their primary chromosome. And they can actually trade these with other bacteria to gain new capabilities like antibiotic resistance. Then you have your plasma membrane, of course, because it is a cell. They have cytoplasm, of course, because they are a cell. They have ribosomes, these little dots, because they are a cell. And then they also have specialized cell walls. Now, not every single prokaryote has a cell wall, but the vast majority of them do. And they might be different based on species, but this just helps them kind of combat water pressure issues, because they're going to be living just in the environment where availability and quality of water will matter, and they don't want to end up taking on too much water or leaking water into the environment. So the cell wall helps protect them from that. Eukaryotes get much more complex. They have their DNA closed up in a nucleus. Then they have a bunch of smaller membrane-bound compartments called organelles, excuse me. So the reason why they're called membrane-bound is because they are made of membrane, or they are surrounded by a smaller membrane. So it's not the plasma membrane, it's some smaller membrane inside. So here's a kind of overall comparison. Of course, there are some that are larger and smaller, but for the most part, you can see here's a virus, here's a bacteria, and here is a part of an animal cell, a eukaryotic cell, with the nucleus right there. Here we see an atom, we have fats, proteins, poliovirus, flu virus, then bacteria is about the same size as the mitochondria. Really small eukaryotic cell is the red blood cell, then animal and plant cells and things like that. Here's a really cool picture. We have a human red blood cell right here, a human white blood cell here, a neutrophil, and this is bacteria that the white blood cell is chasing and trying to eat. So let's dive into some of the organelles. I'm not going to hit these very hard on your quiz. There might just be a little bit of a matching section where you have to match the organelle to its function. Otherwise, I think there's much more important things for us to focus on. So we're going to go through these somewhat quickly. So be able to give me the role, the job of the organelle, and then kind of be able to tell the difference between a plant and an animal cell. There's a couple organelles that plants have that animals don't. So here are your key terms. We're going to start with the nucleus because this is the definition of why this is a eukaryote and not a prokaryote. So the nucleus is the structure that protects the DNA. That's its job. It houses the DNA. So it's surrounded by what we call a nuclear envelope. This is two membranes that surround the DNA. And these two membranes have little holes inside of them called nuclear pores. That way we can get the right kind of enzymes in and the right kind of copies of our DNA can exit. Then inside is something called the nucleolus. Right in the middle is this really dark, dense area, and this is where ribosomes are made. So nucleus, primary job, protect the DNA. Then always next to the nucleus is the endoplasmic reticulum. We just call it the ER because that's much easier. So this is an entirely membrane structure. And as I said, it's always next to the nucleus because the first part, the rough ER, is actually connected to the outer membrane of the nucleus. So as I said, the nucleus is surrounded by two membranes. Well, that outer membrane twists and turns and grows and becomes this rough endoplasmic reticulum and also the smooth down the road. So it's all one continuous membrane structure with the outer membrane of the nucleus. So as I said, there's two different parts to this. There's a rough ER close to the nucleus, and further away there's a smooth ER. So let's talk about the difference between those. The rough ER is rough because it has ribosomes on it. Ribosomes, remember, they're the things that can read the DNA and turn it into a protein. So the main job of the rough ER is to make proteins, and those proteins usually get sent to the outer membrane, so the plasma membrane of the cell. So these are the proteins that are either going to embed in the membrane and become signaling proteins or transport proteins, or even we're going to send them out of the cell, potentially. Then later, we have the smooth ER. This tends to look more like some coral and tubulous coral rather than these flat pancakes. And if we look, there are no ribosomes on it. So this does not make proteins. Instead, it makes lipids or fats, and it'll make all the hormones for the cell in the body, and it also detoxifies the cell. So the smooth ER actually can even act like a little liver. This is where we detoxify toxic chemicals like alcohol and things like that when they enter into our cells. So if we're looking at the body, we have probably, if we're going to go look at our liver cells, because our liver is our detoxifying organ, if we were to look at liver cells compared to muscle cells, we would probably see significantly more smooth endoplasmic reticulum in a liver cell where it has to detox every day versus a muscle cell, which doesn't need to detox quite as much. Then we have the Golgi apparatus. This is an interesting one. It kind of acts, the best description that we can usually make is it acts like the cell's post office. It looks like a stack of flattened pancakes, And what ends up happening is it will receive little packages on one side and release them onto the other side. And in the middle, as these little packages are processed, whatever's inside is processed, we make changes to that molecule, we grow it a little bit more, we add things to it, and when it's finally ready to be sent out, then we release it from the Golgi. So these little packages are called vesicles. They're basically just tiny little membrane bubbles, and they hold whatever protein or lipid or carbohydrate needs to be modified before it can be sent out of the cell. So they come in, they're modified by the post office, the Golgi, then they get packaged, and usually these vesicles will have special markers on them that will direct the cell where to take them, either to the membrane, to other organelles, to the exterior of the cell. It just depends. So there's a special type of vesicle called the lysosome, and this is basically the stomach of the cell. So these vesicles contain digestive enzymes, they contain high levels of acid or reactive oxygen species like hydrogen peroxides, and their whole job is to break down larger molecules that make their way into the cell, whether that be dangerous things like bacteria that need to be killed and broken down, or just food that needs to be broken down into smaller particles before it can be digested and used by the mitochondria to create energy. So usually what happens is the food particles will get swallowed, and then a little lysosome will fuse with the vesicle containing the food, and it'll start to digest it, and then any waste products will get removed, and the food products will get sent back out into the cell to find the mitochondria or be used wherever they're needed to make proteins, whatever that might be. Vacuoles are kind of like lysosomes of plant cells. So for the most part, vacuoles are only found in plants. There's been some weird research that says maybe they're not, but for the most part, we only find them in plants. And their main job is actually to store water, or what we call sap. It's kind of like the blood of a plant, and it helps the plant cell keep the proper turgor or osmotic pressure. So it helps it basically have the right water pressure inside. And we'll talk more about turgor and osmotic pressure down the road. Now, often the vacuole will end up storing dangerous toxins. And this is where if you bite into a plant and the plant is poisonous, you are breaking its vacuole, and those dangerous toxins stored inside you end up ingesting, and you can get sick or die from. So next is the cytoskeleton. So I know it specifies animal cells here. Technically, all cells, even prokaryotes, have a cytoskeleton because it's not membrane bound, but the cytoskeleton looks different in plant versus animal versus prokaryotic cells. When we're talking about eukaryotic animal cells, they have 3 different types of cytoskeletons, 3 different types of proteins that make up their skeletons. And these are really cool. They can be elongated, they can be shortened, they can be moved to different places of the cell, and they offer 2 jobs for the cell, or two roles. They are used for structure, just like we would think a skeleton would be, so they maintain the structure of the cell, and they're used as a superhighway. So those little vesicles don't just float randomly through the cell. Often what will happen is they will get attached to these pieces of cytoskeleton, and they will literally get walked along the cytoskeleton to where they need to go. There's some really cool video animations of this. It looks like a little dude with a big gigantic ball on the back of his, on his back behind his head, and he's just trudging along, walking down the highway here. We also use the cytoskeleton to help during cell division. So this image here is quite cool. We have tagged the 3 different types of cytoskeleton with 3 different phosphorescent or fluorescent coloring, so we can see the 3 different types here in this real image. Then there is the mitochondria. The whole job of the mitochondria is to take food, usually glucose, and convert it into ATP, which is the primary energy currency of the cell. They have a double membrane. They have a regular membrane on the outside and then a more twisty membrane on the inside. They have their own DNA and they have their own ribosomes as well. And we'll talk about what that means in class. So mitochondria are only found in eukaryotic cells. That's a really important part because they are membrane bound. So prokaryotes get their energy in a slightly different way. And then there are chloroplasts. These are in plants only, not animals. They have 3 membranes. So they have 2 outer membranes and then a kind of twisted up inner membrane that forms these like stacks of coins looking structures called thylakoids. And with these ones, they also have their own DNA and their own ribosomes. So kind of interesting. And their job is to take sunlight energy and inorganic carbon, meaning carbon dioxide, and use the energy from the sun to take that carbon dioxide and build food out of it. In this particular case, they build sugar, glucose, and that process is called photosynthesis. So that is it for this video. We'll get into a little bit more detail on the mitochondria and the chloroplast in class, and I will see you guys there.